Hello, my name is Liana Waite, and I'm a PhD candidate in the Department of Ecology and Evolutionary Biology. Today, I want to share with you some research that I've done with financial support from the Centre for Health and Wellbeing. So my research focuses on immunizing the infected. Do underlying parasite infections impact immunization? And today I'm going to focus on the field component of my research, which looks at rabies vaccination in wild raccoons. So just a quick introduction to rabies. Rabies is a viral infection of global importance. It's spread when an infected animal bites another individual, be that another animal or a human, and then it spreads up the nerves of that individual. It's inevitably fatal and it causes encephalitis. So worldwide, dogs are the main vector or reservoir of rabies. But in the US, we don't have dog or canine rabies. Instead, we have wildlife reservoirs. And one of the main reservoirs is the raccoon, which is the focus of my research. So because rabies is such a serious disease, there's a national campaign which is trying to eradicate raccoon rabies via vaccination. This is the National Rabies Management Program. So this program is run or operated in conjunction with the USDA Wildlife Services. Every year since about 1996, they have spread these oral baits, pictured on the right, throughout the northeastern United States. These sachets are coated in caramel to attract raccoons to eat them, and they contain the vaccine. So the idea is that the raccoons will eat these baits and then they will be immunized, and they will no longer be able to spread rabies to other individuals, to domestic animals, or to humans. However, I'm interested in what impacts how individuals respond to vaccination. And we know that not all individuals respond in the same way to vaccination. For example, we know that parasites can impact how an individual responds to vaccination. They can do this either by suppressing the immune system, so an individual is unable to respond, or by pulling, if you imagine a tug of war, as shown here, pulling the immune system in a way that doesn't facilitate immunization. And I actually performed a meta-analysis of this question, do parasites interfere with immunization, which was published earlier this year in Vaccine. And I found that the answer is yes. In general, individuals that have parasitic worms, protozoa, or viruses when they're vaccinated do not respond as well to vaccination as individuals who are not infected. So I wanted to investigate this question in raccoons. And the idea was not only to find out is this phenomenon occurring in this system, but then it would enable me to be able to give some recommendations to the USDA and to the National Rabies Management Program, which might enhance the ability of that program to eliminate raccoon rabies. So this is an important question with practical implications. So I teamed up with the USDA Wildlife Services, who have a longitudinal long-term research station up in Burlington, Vermont. And every year for about the last 10 years, they have trapped raccoons in July and October. So they trap in July to look for baseline antibody levels against rabies. They then distribute the rabies vaccine throughout August, and they re-trap in October to see have the levels of rabies antibodies increased. Have raccoons actually ingested the baits that they distributed in August? So I teamed up with them in 2018 and 2019, and this collaboration is ongoing. And we took several samples. We took blood samples to look for rabies antibodies, and I also made blood smears. I took fecal samples to look for the presence of gastrointestinal worms and protozoa. And we also took some basic demographic and morphometric measurements. So we recorded whether raccoons male or female, were they adults or juveniles, were they lactating if they were adult females, and how much did they weigh and how long were they? So I could make a crude estimate of health, basically a BMI measurement. And as I said, this study was performed in 2018 and 2019. So back at the lab, the fecal samples, to reiterate, were used to look for gastrointestinal parasites. I not only recorded whether they had parasites or not, but I also made an effort to quantify the parasite burden. So do they have just a few worms or do they have many worms? And this was done by counting the number of worm eggs per gram of feces. And then the blood samples were used to estimate whether they had rabies antibodies or not, and also to do blood smears to count white blood cells in order to kind of estimate what sort of immune response the raccoons had when we trapped them. So I'm just going to share some preliminary results with you. The lab work and analysis for this project is ongoing. But a very exciting result is that my hypothesis was confirmed. So we found evidence that raccoons that had worms were less likely to have antibodies against rabies. So if you look, you can look either at the table or the graph, whichever you prefer. But raccoons that had worms were about half as likely to have antibodies against rabies as raccoons that did not have worms. So this is a very strong result suggesting that worms are interfering with rabies vaccination in these raccoons. But this only looks at presence or absence of worms. So then I wanted to know, does it matter how many worms the raccoons have? We could hypothesize that raccoons that had many worms would be less likely to have antibodies against rabies than raccoons that only had 1 or 2 worms. So that's what this graph is investigating. Along the x-axis here, we have the number of worm eggs per gram. So the more worm eggs, we assume means the more worms. And then it's colored by rabies serostatus, whether they have rabies antibodies or not. And you can see there's not really any discernible pattern here. So it appears that nematode burden does not matter. It is the presence or absence of worms rather than the number of worms that is important. And this was supported statistically. There was no pattern. So to summarize these brief results, in wild raccoons, parasitic nematodes do appear to interfere with vaccination against rabies. Raccoons that had worms were less likely to have rabies against antibodies. However, it didn't seem to matter how many worms they had. It's presence or absence that is important, not the number of worms. So thank you for listening. As I said, this work is ongoing, and I have other results that I didn't have time to share today. So if you have any questions or comments, please email me. My email address is lwaite@princeton.edu. Thank you.